Hello everyone, welcome back to our YouTube channel. Today, I'm going to talk about Chapter 4, Biological Molecules. This is for IGCSE CIE Biology 0610 students. I am Dr. Mary, and let's get started. So first of all, what are biological molecules? Biological means for living things. So basically, these are the molecules that are present inside living organisms. So three most important biological molecules for your exam are carbohydrate, lipid, and protein. Carbohydrate is made out of carbon, oxygen, and hydrogen chemical elements, which is the same for lipid. Protein contains carbon, oxygen, and hydrogen, just like carbohydrates and lipids. But it also contains nitrogen and sulfur. So what are the examples? For the carbohydrate, the examples are starch, glycogen, and cellulose. Lipid examples are vegetable oil and animal fats. Protein examples are enzymes, muscle proteins, and hemoglobin. So the subunits. The carbohydrate subunits are glucose. The subunits of lipids are glycerol and fatty acids. The subunits of protein are amino acid. There are 20 different types of amino acids. So you can see in this photo, this is the glycogen molecule. This is one glucose unit. So it means the glycogen molecule is made out of many glucose units. So that's why glucose is called subunits. So there are long chains of simple sugars. So in this example, the simple sugar is glucose. So since glucose is just one ring here, you can see only one ring. That's why it is called monosaccharide. Mono means one, saccharide means sugar. So monosaccharides mean one sugar. So it is a simple sugar. When two glucose molecules are joined together, it is maltose. Since it is two, that's why it is called disaccharide. When lots of glucose molecules are joined together, just like in this example, it is called polysaccharide. So let's look at the fats. So our body mostly contains triglycerides as fats. So the basic units inside the fats are one glycerol molecule, which is the orange one here, which are joined together with three molecules of fatty acids, these yellow molecules. So these four things together, it is called fat. So the fats are actually the solid state of lipids, the solid state at room temperature. When they are liquids at room temperature, they are called oils. So let's look at the proteins here. Proteins are basically long chains of amino acids. Since there are 20 different types of amino acids, one protein may contain different types of amino acids in any order. Let's say like this, if two proteins contain the same type of amino acid and the same number of amino acid, but in different order, those two proteins are different because the order of amino acids are different. So to look at the type of protein, we have to look at the not only the type of amino acids it contains, but also the order of amino acids it contains. So now let's move on to the food test. So these food tests are actually quite important questions for your exam. So I summarized the food test for glucose here for you guys. So this is also called Benedict test because it is tested with Benedict solution. So you have to add a few drops of Benedict solution into the sample, food sample in the test tube, and then heat it in water bath for a few minutes. During that heating time, you will be able to observe the color changes from blue to green to orange to brick red slowly if it contains reducing sugar. So when it shows these series of color changes, it means it contains reducing sugar and the Benedict test is positive. So let's move on to the food test for starch. It is also known as iodine starch test because we have to add a few drops of iodine solution into the food sample to test for starch. If the iodine color, the original iodine color, which is brown or orange, that brown or orange changes to blue or black, blue-black, it means the food sample contains starch and the starch test is positive. Now, let's talk about the food test for protein. It is also known as Biuret test because we have to add a few drops of Biuret solution to the food sample. A positive test means if the color changes from blue to purple or lilac color, it means the food sample contains protein. So let's see the food test for lipid. You mix a few volume of ethanol with the food sample and shake it. After that, you add an equal volume of cold water and shake it again. If the sample shows a cloudy emulsion, it means the test is positive, which means the food sample contains lipid. You have to be able to write this down in your exam paper to gain mark. For if the question asks you to describe whether a food sample contains lipid. So the last food test for this chapter, the food test for vitamin C. You can test whether a food sample contains vitamin C or not by using a few drops of DCPIP solution, which is originally blue in color. So you add a few drops of DCPIP solution into a small amount of food sample that you want to test. If the blue color disappear, which means become colorless, the test is positive, which means the food sample contains vitamin C. So this is all for chapter 4, biological molecules. If you like this video, please like and subscribe our YouTube channel. Thank you so much for watching and see you soon in next video. Bye-bye.